Hello, everyone. The research in my lab focuses around structure and function of the ribosome. So we can view ribosome as a molecular 3D printer. So just like the same 3D printer printed this model in my lab, so ribosome prints all the proteins in each cell of each organism, including our cells. So it is by the action of these proteins, that such important functions as, for example, heartbeat, or oxygen transport in our blood by hemoglobin, or food digestion in our stomach by chymotrypsin. So all these functions are because of the proteins. Essentially, we can look at what ribosome is doing is turning the blueprint of life stored in our genes and our DNA into actual function. So, and not surprisingly, that interfering with this vital process will basically be incompatible with life. So many antibiotics that we use, not like we now, but in general, mankind uses to kill microbes, bind to the ribosome and prevent the growth of bacteria just by inhibiting this vital process. So, but the problem with antibiotic is that no matter how many of those we have, sooner or later they become ineffective because of the developing drug resistance, which is a natural process as all living creatures try to resist every attempt to kill them. So do bacteria and nasty ones as well. Okay, so lincosamides is a particularly interesting class of drugs that binds to the ribosome and is actively used in clinic today. However, there were no new molecules in that class for the last more than 50 years. And recently, in collaboration with Harvard University, so actually they're chemists who developed this interesting drug using a completely new concept of pre-arrangement of the molecular structure. So you can look at this as trying to open the locked door with the keychain, right? So you need to go through all of these keys before you'll find the right one. And the drug that our collaborators invented is like a key that opens the door right away. So not only these molecules are extremely active and potent in a variety of the nastiest pathogens that we tested them against, they also overcome resistance, some of the most common types of resistance. Usually, bacteria become resistant by changing the chemical makeup inside that ribosome. So those orange spheres shown in the picture, basically those are methyl groups that prevent antibiotic from binding it into its natural binding spot. You can look at this as a pushpin on the chair, right? So we can probably sit on it, but it's very uncomfortable. So is the case with antibiotics. In many cases, basically they put like little pushpins so that it's not comfortable for the antibiotic to sit on its chair. But the one that our collaborators developed, engages ribosomes so tightly that actually even with the pushpin on a chair, it still engages it very well and works against drug-resistant pathogens. Thank you very much.